This is Unit 1 of AP Biology, beginning to talk about macromolecules, and a couple things to start off our discussion on macromolecules that are necessary for all macromolecules are these two reactions. The dehydration synthesis reaction, sometimes you'll see it just called dehydration, but I do like to call it dehydration synthesis, and hydrolysis reactions. And so essentially what's going on with all macromolecules, no matter what we're talking about here, is you have the combination of these individual units, or you have these individual units, I should say, they're not necessarily combined, called monomers. And a monomer is just a single unit of a macromolecule. It could be a single sugar or a single or amino acid or anything like that. But you can also have long chains of them here, and a long chain is called a polymer. Really anything that's two or more we could technically call a polymer. Sometimes you'll see the prefix di in front of something like a disaccharide for our two carbohydrates. And so what are these two reactions, dehydration synthesis and hydrolysis? Well, they are how these molecules are combined and how they are separated. So to combine two monomers of macromolecules, water is taken out, and you can see this here. It's water, the H2O is taken out, dehydration, taking water out, synthesis to combine. And so with dehydration synthesis, water is removed in order to combine those molecules. Well, hydrolysis then, hydro, water, lysis, anytime you see that, it means to break something or to break down. And so hydrolysis is using water then to break down macromolecule polymers. So this is whether it's a sugar, whether it's a lipid, protein, doesn't matter. This is how those molecules are broken down. And so first we're going to look at carbohydrates. There are four groups of macromolecules: carbohydrates, lipids, proteins, and nucleic acids. So first we'll look at carbohydrates, the simplest of the macromolecules. Carbohydrates are easy to recognize because they have very regular proportions of carbon and oxygen. In fact, the carbon is always going to equal the oxygens. Sometimes the H is going to be equal to 2 times the number of carbons, but not always. But you will see most of the time it is, as you can see here. All right, and here. And then with glucose, of course, same kind of thing. And so they're easy to recognize. They have the carbonyl groups and the carbonyls, which is the double bonded oxygen, and the hydroxyl groups, which are here. Pretty easy to recognize. For our monomers and polymers of these molecules, the monomer of a carbohydrate is called a mono, makes it easy, saccharide, and polymer then is called a polysaccharide. Now there are lots of examples of mono and polysaccharides. A monosaccharide, for instance, is glucose, one that we'll talk quite a bit about. Fructose is another one. Here's a couple of polysaccharides here, maltose and sucrose, which is a combination of two monomers, two monosaccharides. And notice when they are combined, water is taken out, combine two things, so you have that dehydration synthesis. This glycosidic linkage is the name of the bond that is between two sugars. Carbohydrates will also use the word sugars to talk about carbohydrates. The main function that carbohydrates serve in living things is energy. They are an easy energy source And most living things will gravitate towards carbs before they will gravitate towards other types of energy because they're easily broken down. Carbohydrates are also stored in plants and animals. Plants store carbohydrates in a molecule called starch. We're all familiar with starch with like potatoes. Well, potatoes nothing more than that plant's storage of all those carbs that it's taken in from and made from the sun. Um, Glycogen is the same kind of idea, but with glycogen it is animals instead of plants. So glycogen is how animals store carbohydrates. A couple other carbohydrates that you need to be familiar with. One is cellulose. Cellulose is a structural carbohydrate found in plant cell walls. It is the most abundant compound, organic compound found on Earth. Just because of that, it's all over the place. Interesting, the difference between cellulose and starch is very minimal. It has to do with just a single bond structure, but that single bond structure makes it difficult, for instance, for us to break down cellulose, but we break down starch very easily. So another type of carb that you need to be familiar with is chitin. Chitin is a carb that's also a structural carbohydrate. It's found in the exoskeleton of many animals, um, like arthropods, for instance. It's also found in fungal cell walls. Here you can see this surgeon using chitinous thread in order to sew up the person that they're operating on. So there are other applications for chitin. It's a very strong carbohydrate. Next, we'll look at lipids. Lipids are things like oils and waxes and steroids, fats up here, oils and fats and steroids and waxes. The primary function of the lipid is to store energy. They do have other functions, but that will be the primary function that we are going to be looking at. Here's a picture of a basic lipid. The monomer of lipids is called a fatty acid. As you can see here, this structure here is known as a fatty acid. It is a long chain of hydrocarbons. And remember, we talked about hydrocarbons being a big source of energy. And you can see here dehydration, synthesis, water being taken out, this fatty acid being joined to this molecule. Now, fatty acids are a little bit different in that they're not joined together necessarily, but they are attached to this molecule here that is called a glycerol. And so, just as a real simple drawing of this, you have a glycerol molecule here, and you might have 3 fatty acids connected to it. And so each of those fatty acids are the monomers, and this whole thing would count as a polymer. Here's an example of that polymer. Triglycerol molecule, or maybe a word you've heard before, is a triglyceride molecule. And if you've ever had like blood work at the hospital, they'll tell you what your triglyceride count in your blood is. It's just the amount of fat in your blood. The bond that connects a fatty acid to the glycerol is called an ester linkage, so you should know that. There are a couple different types of lipids that we're going to look at. The triglyceride is very common, and then even on the triglyceride there are different kinds of fatty acids. Two most common are the saturated and the unsaturated fat. Saturated fat is a fat in which there's no more hydrogen that can be added to it. It is completely saturated with hydrogen. These, and I'll show you, here's a picture of some saturated fats. Notice you can't add any hydrogen to any of these. They're completely full. They are typically solid at room temperature. They're associated with animal fats, but not always. Um, Sometimes these are called bad fats in our diet, but you need saturated fats in order to survive, so they're not altogether bad, but they can be in excess, just like just about anything. The unsaturated fat is called unsaturated because of double bonded carbons. So I'll draw a picture of this. So whereas with the saturated fat, you have carbons that are completely full of hydrogens, with the unsaturated fat, you have these carbons that could have more hydrogens if, I'm not going to fill in the rest of those, you get the idea. They could have more hydrogens if those double bonds weren't there, and so they're considered unsaturated. Now, we have ways of saturating those unsaturated fats. Those unsaturated fats are typically liquid at room temperature and are associated with plant oils, like here, this corn oil picture here. We do have ways of saturating those unsaturated fats that are typically called hydrogenated fats. Hydrogen has been added to them, or trans fat, which is a very unhealthy option. Another place where lipids can be found is in cell membranes. Phospholipids have them. Phospholipid here with the hydrophilic head and the two hydrophobic tails. You see a fatty acid here, an unsaturated fatty acid there. See, there's the double bond and the carbon. There's a picture of the cell membrane. Waxes are nothing more than a fat plus an alcohol. They're able to do a lot of different things with waxes. Candles obviously are one of the options there. And then steroids. Steroids, they are always look like this, or at least this portion of the steroid. They have these 4 carbon rings. This is the variable structure that makes one steroid different from another. Here's a picture of that as well. You see these 2 different steroids that are reproductive hormones in male and females, but they have that same base structure of those 4 carbon rings. And so steroids function in lots of different ways, but they mostly function in their signaling hormones in the body that tell cells to do things, send messages from other parts of the body. One important hormone in your body is cholesterol. I'll zoom in to show you this, but you can see the cholesterol there inside the cell membranes. Cholesterol actually builds up and helps with cell membrane flexibility. All cell membranes do need to have some of this, but any, of course, cholesterol in excess is bad for your health. And when we have excess of lipids in our system, we build that up with tissue that is called adipose tissue, or just fat tissue. Of course, in the animal kingdom, fat is good because of their lean times, but now that we have things like grocery stores and McDonald's, there aren't really lean times for most of us, and so adipose tissue can become a problem.